Greetings again, Biology 101 fans. Today we're going to be doing a different topic from Chapter 4 in the Mater Biology 101 manual. It's on diffusion and osmosis. It's from Section 4.3 in the manual on diffusion, Section 4.4 on osmosis. To begin with, let me just introduce you to diffusion a little bit and give you a kind of crude demonstration. Then we'll do one of the exercises from the lab manual. So the thing to remember is that diffusion is constantly occurring, and diffusion is really just the slow movement of one substance through another. So I've got that written out here in a piece of paper for you. Diffusion is just one substance moving, usually kind of slowly, one substance moving through another. It could be a solid, a liquid, it could be gases. Generally, you're thinking in terms of liquids when you think in terms of biology, but there are some solid-type phases too, and certainly gas for gas exchange and so forth. Diffusion is always resulting from the constant random motion of particles. Whatever particles you want to think of on the atomic level, ions, molecules, and so forth. But they're always bouncing around. And to give you a quick crude demonstration of that, what we mean by constant random motion, here are some little bugs they're called from a kid's toy that sort of bounce around in these chambers. This is illustrating just what we mean by constant random motion. They start out in one chamber, but just by bouncing around, they will eventually migrate or diffuse into a different chamber. That's all you really mean by constant random motion and diffusion. So to get to one of the exercises from the manual, here I have two Petri dishes set up. This is a Petri dish with some agar in it. Agar is a kind of starch that's derived from algae, and when you boil it up in water, it forms this kind of a semi-solid material. And in this hole, I've placed some dye. I inoculated the dye in there early this morning, so it's been sitting there for about 5 hours now, and it's diffused. You can see the dye just sort of spreading out through the agar, semi-solid material, and that's been diffusing for 5 hours. So I'm going to measure the distance that's diffused here with a ruler. I'm going to measure from the edge of that hole to the very edge of where it's diffused. I'm going to call that 11 millimeters. So in the solid here, it has diffused 11 millimeters in 5 hours. Again, now this is going to be difficult to read, I know, on the video, but I will summarize the data for you in a table and post it so that you have access to that, and it's easy to see all the numbers. All right. Now, in this other Petri dish, I just have some distilled water. And I'm going to put a drop of the dye in the distilled water and time to see how long it takes to diffuse, or measure how far it diffuses in, let's go for like 3 minutes. I'm going to be aiming for that origin of the ruler right at 0, but this will actually diffuse, or it will scatter over the surface pretty quickly. So what we want to see is this dye moving from that initial place. I'm going to look at here and say it's like 5 millimeters right here. And in a few minutes, we'll come back and see how far this spot has diffused. We're not worried about the dye scattered along the surface and so forth. That's just some strange solubility effect. But we're going to take it at 5 millimeters to begin with. Let it go for a couple of minutes, and then measure the position again. So for the start here, it's at 5 millimeters, and we'll see how long it diffuses, how far it diffuses in maybe 3 minutes or so. So we'll be back in about 3 minutes. So we're back, it's just about 3 minutes. I'm going to look at here, and I'm going to measure, it's kind of scattered, but I'm going to say this is at 16 millimeters. Start out at about 5. It's really moved quite a bit, so I'm going to say 16 millimeters. So at the end here, it went to 16 millimeters. So if we do the subtraction, initially 5, 16 at the end, that's going to be 11 millimeters that has actually diffused in 3 minutes. So in 3 minutes, in the liquid, just liquid water, it's diffused 11 millimeters. So again, I'll have this all posted on in a convenient table for you later. Next, we're going to do some work on diffusion through a plasma membrane. So next section here is going to be still diffusion, but now we're going to add this variable called a plasma membrane, or cell membrane. The manual tends to call it a plasma membrane, but just think of it as a cell membrane. So the important points to notice here is that any kind of cell membrane or a plasma membrane is selectively permeable. Permeable simply means that a material can pass through some barrier or some border like a cell membrane. Selectively permeable means that some materials can pass through the barrier, but others cannot. So a really good example to keep in mind is the screen in a window. The screen is permeable to air. Air can pass easily through the screen, but hopefully the bugs like mosquitoes can't. So it is not permeable to mosquitoes or bugs. Therefore, the screen is then selectively permeable. Same thing happens with a membrane, a cell membrane. It's just a lot more sophisticated because lots of things can pass through by different means. To do a simple example of semi-permeability too, I have here a piece of mesh. And what we're going to do for the experiment here, the actual experiment, is use this piece of dialysis tubing. Here's a piece of dialysis tubing. It's a kind of plasticky type material, and you have to keep it wet so that it stays pliable. But this has pores in it. So I'm going to put a couple solutions in here, and the pores will let some things pass through, but prevent other things from passing through. So to give you a feeling for how this works, here is a piece of nylon mesh. The holes in the nylon mesh are really going to be similar to the pores in the dialysis tubing that's been tied into that bag. And here I have just a mixture of some sand and some little white chunks. These are called boiling chips. So if I simply pour this over the nylon mesh, let it bounce around a little bit, you can see that the sand passes quite easily through the nylon mesh, but the boiling chips, the white chunks, don't. So this mesh is going to be selectively permeable too, obviously just because of size. The sand particles are easily getting through this because they're small enough, and the boiling chips are just too big. Obviously, cell membranes will prevent or allow stuff to go through them based on things other than size, and that's what you can deal with in your lecture section. But for now, we're just using this dialysis membrane. This is a simulation of a cell membrane, but again, it's mostly just a matter of size, because this again has pores through it. It can either let stuff pass or not. So I'm going to put two different solutions in here. I have some starch solution. I'm going to put that in there, and I'm going to have some glucose. This is a relatively concentrated solution of glucose. The sugar, that's going to go into the bag too. And in the bag, the dialysis bag here, I'm going to put in a beaker with some water. And then we're going to analyze whether or not the glucose passed through, whether or not the starch passed through, or what exactly is going to be going on in here. So we're going to have to analyze for glucose and starch too. In order to do that, we're going to have to use some of the chemical tests you've already gotten used to. So let me just remind you of the chemical tests you've already seen. In this tube, there's a little bit of starch. It tends to settle out in the bottom of the tube here because it's not entirely soluble in water. And to that starch, I'm just going to add a drop of iodine, because this is the class test for starch from your chemical composition lab. I'm sure you remember really well, right? So here is a drop of iodine into the starch, and remember what happens when you mix the starch with some iodine. You get this black color developing, or a darkish color. This is fairly dark, kind of a little bit of a purple grayish cast. And there's some particles in there, if you can see that in the camera too. That's the starch, again, that's not entirely soluble, but absorbing the iodine and just changing color. So that's the starch test with iodine. The other test that I won't demonstrate immediately, but the other test we're going to use is for glucose, and this is Benedict's solution. Benedict's solution is this light blue solution here. You put something in there you suspect it has some sugar in it. You boil it up because it has to be heated so the reaction occurs, and you get different shades of colors. It starts out as this light blue. If there's some sugar present, it could turn greenish or yellow or orangey depending on how much sugar is in there. If there's a little bit of sugar, it will turn green. If there's a lot more, they'll turn yellow. If there's a significant amount, it could turn orange or even red. So that's how we're going to analyze for the sugar too. So in this beaker here, I'm going to put some distilled water. And again, I'm going to take this piece of dialysis bag, this tubing tied into a bag here, and put in some glucose and starch. And then we're going to let this sit in the water here. So I have 2 tubes ready for Benedict's test. BT at the top here stands for Benedict's test at the start of this particular experiment. I'm going to take some solution out of the beaker right away. And then the other one is labeled BT at the end. So for Benedict's test at the end of this procedure. So I'm going to put in some glucose solution first. Fill the bag about halfway full with glucose. And then do the same thing with starch. The starch, again, is kind of whitish, so that means it will look different. And you can see here the starch and the glucose mixed into this bag. And all I'm going to do is going to put this bag into the water, a beaker full of water, secure the top here with a rubber band. And so this is going to sit here. I'm going to time this. I'll put on a timer. It's going to sit for probably 15 minutes or so. The time is not crucial. But in the meantime, before this gets very going, I'm going to take some of the solution of the beaker out, and I'm going to put it into this test tube that says Benedict's test at the start. So that's our sample of the beaker water here before much diffusion happens. And we'll just see what happens at the end of this time period. In the meantime, let's record what this stuff looks like before diffusion. Oh, so the other thing I'm going to do is put in some iodine. I'm going to put some iodine into the beaker water so that it has that nice iodine color. So you can see here, it's got iodine in there. We'll see what happens with the iodine in the beaker too. But for the moment, I'm just going to record what the stuff looks like. In the bag, at the beginning, the appearance of the starch is whitish from the starch in that bag, and you can still see that. You can see a little bit of the white there from the starch in that bag. And then we're going to record the appearance after this has been sitting around for a while too. We're also going to record what the water in the beaker looks like. Here is a place to record this. I'm just going to call it light brown color because that's the color from the iodine. And again, we're going to look at that color in about 15 minutes. We'll be back when things change. So we're back after this has been, the bag has been sitting in the beaker for almost a little over 15 minutes now. So let's take a look to see what it looks like here. I'll give you a close-up. Here is the bag in the beaker. Notice the bag has got a fairly dark color near the bottom there. It's no longer just whitish. And then again, we want to look at the color of the water in the beaker. That still looks relatively uniformly brown. The light brown color again from the iodine. So let's go ahead and record that, and then we'll do a Benedict's test. So on the bag, at the end of this incubation period, when things have diffused a bit, we see that it has a blackish color in it. It's not uniformly black, but obviously that's the thing that's really changed here, is that there's a significant black color in the bottom of the bag. You can still see, I can still see some of the white in there too, but there's quite a bit of black all around the edges there. Now for the beaker, again, the beaker doesn't really look like it's changed in appearance very much again, so I'm just going to record a light brown color here for the beaker, before and after, start and finish. Next thing we're going to do is we're going to do a Benedict's test on the solution. Because again, remember that Benedict's test is going to be testing for the presence of a sugar, in this case glucose. Here's the tube we're going to use. I've got it labeled BT for Benedict's test, and it was Benedict's test sampling the beaker solution at the end of this incubation period. So I'm just going to withdraw some of the beaker, some of the water from the beaker, put it into this tube. That's what we're going to use for a Benedict's test. Then here is the tube with the beaker solution for Benedict's test at the beginning versus the Benedict's test from the beaker at the end. You got all that? All right, so to do the Benedict's test, remember we have to cook this stuff. We got to heat it up in boiling water. Here's a boiling water bath. Pretty close to boiling. I'll give it a little boost here. We're going to let that sit for about 5 minutes or so and then come back and take a look at those tubes. So in 5 minutes we'll be back with the end of the Benedict's test. So here we are after a few minutes of the Benedict's solutions cooking away in the boiling water. And notice they really do look different. Here is the tube with the beaker water at the very beginning of the incubation period for the diffusion. So this is Benedict's test at the start. Guess I should give you this view. Benedict's test at the start. And let's look what it looks. It's blue, right? It looks like Benedict's solution. So I'm just going to record this at light blue color. And if we look closely, it's still fairly clear. You can see the lettering through the solution here. It's clear. There's nothing solid floating around in there. So I'm going to write "and clear." By contrast, let's look at the tube at the end of the incubation period. Here, this tube is obviously not blue anymore. It's kind of reddish. And if you look closely, you can see some chunks floating around in there. Well, it's probably pretty hard to see in the video. In any case, it's a very different color. So I'm going to call this, well, you can see some of it starting to settle out here. There's a little bit of extra red at the bottom. If I don't mix this, it's settling out. And that's a solid material. We call that a precipitate. So it's a reddish color. And clearly solid material in there that's floating around and settling out. And we call that a precipitate. Precipitate is something that comes out of solution, a solid that comes out of solution. But notice this looks pretty different than the Benedict's test at the beginning. You can make conclusions about what that must mean. We're going to do some stuff on osmosis next. So in this apparatus, I've got yet another piece of dialysis tubing tied into a bag, noted at the end, tied into a bag, and then I tied it onto the end of this glass tubing here. Same kind of dialysis tubing, and then I filled it full of molasses. And the molasses is basically a sugar syrup. So this is a demonstration of osmosis. So let me give you a little blurb on what osmosis is. Osmosis is really a special case of diffusion. Osmosis is the case of just when water is going to cross the cell membrane. Water is always going to be some component of cells somewhere. It will always be able to cross cell membranes, more or less fast or slowly, whatever, but it will always be crossing the membranes. In some cases, solutes cannot cross the membrane because they're ionic or maybe just too big, and in that case, it's the water that's going to be crossing the membrane rather than one of the solutes. Sometimes the solute is just going to be crossing very slowly, but then the water crossing becomes more significant because it's just that much faster. So in this particular demonstration, again, we've got molasses in this bag, and you can see that it's being pushed up this glass tube in the inside. If I lower the tube so the molasses is actually sitting in the distilled water in the beaker here is full of distilled water, and there's a little bit of brownish color just because some of the molasses has been seeping into it. So there's just distilled water in the beaker. Again, the molasses is in the bag, that's why it's brown. And molasses is just a really concentrated sugar syrup. So there's an awful lot of sugar in there. And what I'm going to do is I'm going to measure the position of the molasses above the edge of the tube. I'm going to measure it now, and then I'm going to measure it again in about 10 minutes. And we'll calculate a rate of how fast the molasses is getting pushed up the tube. And this, again, is due to osmosis. So I'm going to measure this position at, it's going kind of fast here, 61 millimeters. So the initial position is 61 millimeters. At time 0, come back in 10 minutes, we'll measure it again, see how far it's gone. So we're back after 10 minutes. I'm going to measure the position of the molasses again, but notice how much it's migrated up this tube. This sucker's really screaming away here. So I think I got a long enough ruler. On the edge here, some water on the top. I guess I'll take that. I'm going to call it 242 millimeters. So at the end of 2 minutes, the position is 242 millimeters. So 242 millimeters minus the 61 initially. See if I can do the arithmetic in my head. This is 18, so 181 millimeters it's traveled. So this is the total distance traveled. Again, I'm going to put this in a table for you. Everything will be summarized in a nice, neat fashion for you. So the rate in 10 minutes would be equal to 181 millimeters divided by 10 minutes. And you can do that arithmetic. Next up is one more osmosis experiment using elodea leaves, an aquatic plant. So for our last exercise in osmosis, this is another version of osmosis. We're going to use the aquatic plant we have in the lab here called elodea. There's a little bit of sprig of elodea here. I'm going to rip off some leaves here and put it in different solutions. So I'm going to put it in distilled water. I'm going to put it in some sodium chloride solution. And you can see what happens to the cells when they're in that environment. The key term here is tonicity. Tonicity refers to relative concentrations of solutes and water, for that matter, on different sides of a cell membrane. So inside a cell, outside of a cell. So it's going to compare two things. It compares the concentration of the solute inside the cell and outside the cell, and it's going to compare really the concentration of water outside a cell and inside a cell. This is another example of osmosis too. Tonicity is a very important concept in medicine. You'll see it quite a bit if you move on from Biology 101. In any case, what I'm going to do is expose these leaves again to either high concentration of sodium chloride and then some distilled water. So there's three other pieces of terminology you need to remember here. Hypotonic means that the solute concentration outside a cell is lower than the concentration of that solute inside the cell. Isotonic means the solute concentrations are on both sides of the cell membrane, inside and outside. Hypertonic, it's really similar term here. You just got to keep them straight. Hypotonic versus hypertonic. Hypertonic, hyper means more. Hypertonic means that the solute concentration outside the cell is greater than the concentration of that solute inside the cell. Okay, so I'm going to use two different concentrations of sodium chloride. This one's going to be 0 because it's just distilled water. This one's fairly high concentration. It's 10% sodium chloride. 10% by weight. So in other words, if you've got 100 grams of solution, 10 grams of that 100 is sodium chloride. Fairly high concentration. So I'm going to put this Eladia on these microscope slides. We're going to take a look at them in a microscope. We've got a camera in one of the oculars on a microscope, and so I can show you photographs. Hopefully I'll show you a photograph of what these leaves look like when they're in these particular conditions. So I'm just going to rip off one of these little leaves of Eladia and put it in one case in a drop of water. See if I can get this to lie flat. And put a cover slip on it. And then the same thing on this slide. Take a piece of Eladia, little leaf. But in this case, I'm going to put it in sodium chloride. Put a drop of that on the slide. Put the leaf in the sodium chloride, and then let it sit for a few minutes. Get a cover slip on this one. Then I'm going to put these on a microscope and take photos, hopefully, of a microscope so you can see the difference. What the cells look like in the presence of 10% sodium chloride and in just distilled water. So then you're just going to, there's no video there, it's just going to be photographs. You can decide which one is going to be in sodium chloride, which one is going to be in the distilled water. And no doubt your instructor will have you with that one too.